bacterial growth curve. Now, before I discuss the bacterial growth curve, we need to understand an important term, that is generation time or the rate of growth, which means the time taken by a parent bacterium to produce two daughter bacteria by binary fission. Alright, so it is the time taken by a parent bacterium to produce two daughter cells via binary fission. This is the definition. The generation time or the rate of growth is constant for a specific type of bacterium. Okay, it cannot be changed. I'm going to take the example of number 20. Okay, 20 is an easy to remember number. To understand the significance of generation time or the rate of growth. So, remember 20 minutes, 20 hours and 20 days. Now, these are the three examples of generation times which we are going to understand. In case of bacteria where the generation time is around 20 minutes, it could be 18 minutes, 18, 15, 20, 22, 25. So, basically roughly around 20 minutes. What will we observe? If we put those bacteria on culture media, we will observe growth or colonies the next very day on the culture media. Okay, so we can get growth of the bacteria, which we call as colonies on the culture media, colonies on the media within a day. So, very rapidly growing bacteria. So, this very rapid generation time is encountered in a lot of common organisms. For instance, Staphylococcus, alright, Escherichia coli, Klebsiella. So, if the patient sample has got any of these bacteria whose generation time is within minutes, next day only we can see the colonies. Now, as far as 20 hours is concerned, it means one bacterium 20 hours from now is going to give rise to two daughter cells, and those two daughter bacteria further will give rise to their own daughter cells after a further span of 20 hours. Okay, so this is much longer time period required in comparison to 20 minutes. And we have got a very famous bacterium, Mycobacterium tuberculosis, which we need to remember in this context. Its generation time is not exactly 20 hours. It is somewhere between actually 13 to 14 hours. Okay, that is why, if we are suspecting MTB in the clinical sample of a patient, we should incubate the media for a period of 6 to 8 weeks. Okay, we should wait for 6 to 8 weeks incubation period, because that is the time range within which the colonies are expected to appear on the culture media. So, the colonies appear in 6 to 8 weeks time. Alright, so they are a little slow growing bacteria. Now, who is the slowest of all the bacteria, all the medically important bacteria? 20 days. 20 days, such a long generation period or the rate of growth is encountered in Mycobacterium leprae, the causative agent of leprosy. Okay, now imagine there is a patient who observes some kind of skin lesion and he's very petrified. He becomes very afraid. He wants to rule out leprosy. Okay, so he runs to the dermatology OPD and he meets the dermatologist. What do you think the dermatologist will suggest? What will he tell the patient? That you can, we'll collect your skin sample, we'll send it to micro lab, and those people will culture it, and you come to me after 6 to 8 months, and then on the basis of report, I'll tell you whether you have leprosy or not. Okay, that doesn't happen, because if 20 days is the generation period, the colonies are going to appear in 6 to 7 to 8 months. Do we, should we wait for all these for such a long period of time to wait to find out whether the patient has leprosy or not? Definitely not. So, remember, the culture of Mycobacterium leprae is clinically irrelevant. We do not do culture for diagnosis of leprosy. This is one problem, that this organism has got such a long generation period. And guys, the other problem is we anyways do not have any culture medium to grow this bacterium. Okay, even if any proper culture medium was available, then also the 20 days generation period would have been a big problem. Okay, so that is why the culture of M. leprae is clinically irrelevant. So leprosy is a microscopic diagnosis. We try to observe the presence of these bacteria under the microscope by doing Zn staining, and there are some other methods of diagnosis also, but not culture. Okay, so remember, the culture of this organism is clinically irrelevant. Alright, so with this understanding of the generation time or the rate of growth, now let me start the topic per se, that is the bacterial growth curve. Alright, so guys, when we are going to talk about culture media in bacteriology, I am going to tell you that culture media in the simplest, in simplest terms can be understood as food for bacteria. Okay, we are literally trying to bribe the bacteria which are probably present in the clinical sample of the patient. We are trying to bribe the microorganisms by putting the sample of the patient on culture media, which has got a lot of nutrients, right? In the hope that if any organism is there in the sample, it should be able to grow. Alright, so culture media can be of, broadly we can categorize them as solid media, semi-solid and liquid media. Now as far as the liquid culture media are concerned, the liquid culture media are also known as broth. Okay, so suppose this is a test tube and it has got this liquid culture media. This is a broth, liquid culture media. Alright, and suppose we add certain bacteria, you add some bacteria to this test tube. Now what you are doing, actually you are exposing these bacteria to favorable conditions. Obviously culture media is a favorable condition for the bacteria. So what do you expect? You expect the bacteria to grow or multiply, right? So now we are going to see how the growth of the bacteria will occur, and we are going to understand this by plotting a graph. Okay, on x-axis we have got time, and on y-axis we have got number of bacteria. Suppose we add to this test tube, suppose we add 5 bacteria. Okay, at time 0 we now just added 5 bacteria we added. Okay, so what do you think? Should the bacteria immediately begin to multiply? No. Why not? Even though the culture medium is said to be favorable. The reason is, guys, whenever the environment changes, okay, whenever if we take the example of humans, whenever our environment changes, we take a little time to adjust. Okay, for instance, when you people go from hostel to your house, now going to your home from the hostel is obviously going to a favorable environment. Alright, okay, so for most of the students, homes are more favorable than hostels. There will be a little some exceptions to this general rule which I am saying. Okay, so my point is, once the students go to a favorable environment, for instance their homes, they still will take some time to adjust. They'll be really happy. Okay, once suppose they're coming to their house from the hostel today, they will initially oversleep. Okay, they would like to have they would like to spend a lot of time eating, going out and all. So they will initially be very happy. Okay, and then gradually they will start the other activities. Similarly, in case of bacteria over here also, even though you are exposing the bacteria to favorable environment, that is culture media, initially the bacteria will accustom or adjust themselves to this favorable condition. Okay, so initially there won't be any increase in the number of bacteria. Okay, so this is referred to as the lag phase. Okay, lag phase. So we started with what? We started with 5 bacteria in the test tube. For some period of time, the number will not change. It will remain 5 only. This is the lag phase. Then basically during the lag phase, the bacteria are switching on their enzymes which they would need in future for multiplication and for their metabolic activities, etc. So once they have done all this preparation, they will enter the second phase, that is the log phase or the exponential phase. Log or the exponential phase. Okay, so as we can see, now the number of bacteria is going to multiply. All right, but as time passes, what is going to happen? The number of bacteria is going to be more and more, but the culture medium in the tube is limited. The culture medium is not going to increase. All right, so the culture media ultimately will not be enough for the increasing number of bacteria. So there will come a point when some bacteria will be devoid of the nutrients and they will begin to die. Okay, so after the log phase, the bacteria will enter what is known as the stationary phase. Okay, in stationary phase, what happens? The number of some bacteria begin to die, and the number of dying bacteria is equal to the number of living bacteria. So the bacterial number again becomes a constant, right? Then as time passes by further, what has happened? There's going to be death of even more bacteria. There will be accumulation of metabolic waste products by the bacteria also as a result of their metabolic activities. So more and more bacteria now will die and less will survive. So next what will happen? The next phase will be encountered, which we call as the phase of decline. Phase of decline. Alright. Now, this is referred to as the bacterial growth curve, and some important questions are asked just out of this curve. For instance, when does sporulation or spore formation begin? So, spore formation already we have discussed before. So, spore formation should take place when the conditions become unfavorable or adverse. And where is it happening? It is happening in the stationary phase. Alright. So, remember S for stationary, S for sporulation. So, it is in the stationary phase that sporulation starts or spore formation starts. Alright. So, the next question is, in the log phase, the rate of growth is constant or increasing or decreasing. Okay. Now, think about this. The rate of growth, what do you think? Constant, increasing or decreasing? Now, I get the answer from many students to this question as the rate of growth is increasing. No guys, it is the number of bacteria which is increasing in the log phase and not the rate of growth. Remember, at the outset only I had clarified that rate of growth or generation time is going to be constant for a specific bacterium. Alright, it is the number which is increasing during the log or the exponential phase. The rate is constant. Okay, so the rate of growth in the log phase is constant. Alright, then there's another question. When does the rate of growth begin to increase? Okay, so basically, in other words, when do the bacteria actually begin to multiply? When does that happen? It happens over here. Okay, so at this point of time, here the rate of growth increases. The bacteria which were previously not multiplying while they were in the lag phase, this is the lag phase, and once all the preparation of enzyme production and everything else which the bacteria need for multiplication, all that homework has been done by the bacteria, they will jump into the log phase. So at this interface between the lag phase and the log phase, we say that the rate of growth increases. Okay, the rate of growth increases, or in other words, the bacteria begin to multiply. Alright, then also at this particular point of time, that is at the interface between the lag phase and the log phase, the bacterial cell size is maximum. The bacterial cell size is maximum. when does the maximum amount of endotoxin produced? In which phase of the bacterial growth curve? That's another question. Now, endotoxin also we have discussed in bacterial cell structure or anatomy. Now, we all know that endotoxin is a part and parcel of the structure of gram-negative organisms, where it is located in the outer membrane, alright, and it's an integral structural component. It is going to be released only on the death of gram-negative bacteria. Now, in this entire curve, when do you expect the maximum amount of endotoxin to be produced? When the maximum number of cells are dying, and that is happening, guys, in the phase of decline. So, in the phase of decline, maximum amount of endotoxin is produced. Maximum amount of endotoxin or LPS is produced. Alright. Then in the bacterial virulence factors, we are going to talk about exotoxins. Now, exotoxins are proteins. Now, remember, the proteins are also a product or a result of metabolic activity. And when is the metabolic activity maximum? Remember, the metabolic activity is maximum. The metabolic activity is maximum in the log phase, that is the exponential phase. Okay, this can explain that the maximum amount of exotoxin, which is also a protein and hence a result of metabolic activity, the maximum amount of exotoxin should be produced in the log phase. This is as per the latest articles, but most of the standard textbooks, they stick to the stationary phase as the answer. Okay, so let us stick by the books only. So the maximum amount of exotoxin is produced during the stationary phase. Alright, it is produced during the stationary phase. This is again a schematic representation of the bacterial growth curve. So that's about the bacterial growth curve, guys.